students, welcome back. We're going to continue with our discussion of the formed elements. In the last lecture, we looked at the structure of these formed elements and their appearance. In this lecture, we're going to take the images that we created in the last lecture and annotate them. We're going to also describe the function of these formed elements, and we'll give the names of these formed elements. Let's begin. Our first formed element is shown here. It is a red disk. So let's discuss this one. This red disk is the most abundant formed element in the blood. If we were to draw it on its side, it would look like this. First off, we can see that this cell is round like a disc. So this cell is a disc. And also, looking at the side view, we can see that there's a concavity here and a concavity here. And because this cell is a disc with two concavities, it's referred to as a biconcave disc. So we're going to write that here. The main function of this cell is the transport of oxygen. So we'll put here function. This cell has no nucleus. And this formed element is a red blood cell, or an RBC. And this is our first formed element. Let's next describe our second formed element. This one here. This formed element is known as a cell fragment, and it's involved in clotting or coagulation. So let's indicate those here. And this formed element is a platelet. We've seen this one already. Before we start describing the other formed elements, let us give a general description of the two groups. The two formed elements on top have large nuclei and contain no visible granules, whereas the ones on the bottom, the nuclei are lobular, they have lobes, and they contain granules. And so the general name for the group on the bottom is going to be the granulocytes, or the cells that have granules. And the two cells on top, because there are no visible granules, we call these agranulocytes. Keep in mind, granules are present, however, they just can't be seen with a light microscope. And when these cells were first discovered, they were discovered using a light microscope. So these are the agranulocytes. And so these are the two names of these groups of cells. The agranulocytes, which contain no granules, and the granulocytes, which do contain granules. Okay, let's continue with our specific descriptions. Let's start with this cell here. Let's first just describe what we see with this cell. Most obviously, there's a very large nucleus, a very thin rim of blue, clear cytoplasm. You can see that. And so let's indicate those two first. There are three groups of these particular cells, and each has a variety of functions, and some of these functions overlap, but I don't want to get into those specifics right now. So I'm just going to give two general functions of these cells. The first is that they destroy cancer cells, cells infected with viruses, and foreign cells. That's the first function. And the second function is that they secrete antibodies. So let's indicate those two here. So this is a general function of the entire group of cells. Now this next function is really specific to one of these, and this is the secretion of antibodies. One group secretes antibodies. So let's indicate that here. And now for the name of these cells. These cells are the lymphocytes, and there are three groups of lymphocytes: the B cells, the T cells, and the natural killer cells, or the NK cells. We're going to indicate the lymphocytes and those three names here. Let's describe our next group of cells. First, let's just describe what we see. These cells have a light violet nucleus and an abundant cytoplasm, so let's indicate those. These cells function in phagocytizing pathogens and dead neutrophils, so we're going to indicate that here. And now for the name of these cells. These cells are known as monocytes when they're in the bloodstream. However, when they move into the tissues, they're called macrophages, and they can become huge then. And so we're going to indicate those two names here. And this describes our fourth formed element. Let's move to our fifth one, our first granulocyte. First thing we're going to do is describe what we see. First, the cell has reddish violet granules, and this cell is also multi-lobed. The nucleus may have from 3 to 5 lobes. We're going to indicate those two here. These cells function in destroying bacteria. So these cells destroy bacteria. We're going to indicate that here. These cells are known by two names. They're called neutrophils, and they're also called polymorphonucleocytes, or polymorphonuclear cells, or PMNs. So we're going to indicate those here. And so this describes our fifth formed element. Let's move to our sixth one. Let's first describe what we see. This cell has orange to pink granules, and the nucleus has two large lobes. These cells function in fighting parasites and allergens. So let's indicate that. These cells are known as eosinophils. And that completes our description of the eosinophils. Let's move next to our last formed element. Okay, first let's describe what we see. These cells have large granules, and we will indicate what's in these granules. Some of these granules have anticoagulants, and we'll name those, and some of these granules have vasodilators. And these cells have a large nucleus that's obscured by these granules. Let's indicate the large nucleus first. It can be U-shaped or S-shaped. And these cells also have large granules that some have anticoagulants and some have vasodilators. These cells exist primarily in the tissue. And so if there's ever an infection in the tissue, the anticoagulants make sure that the blood doesn't coagulate so that the other white blood cells can get to the site to help. And the vasodilator makes sure that the blood vessels don't close so that the other white blood cells can get to the site. So we're going to indicate those here. As I mentioned earlier, the large granules include anticoagulants and vasodilators. The anticoagulant we've seen already is heparin, and the vasodilator is histamine. Let's indicate those two. And these cells are called basophils. They're basophils when they're in the bloodstream. However, when they move into the tissue, we call them mast cells. Let's indicate those two here. And so this concludes our description of our last formed elements. And so let's summarize all of our formed elements. First, we have a red blood cell. This red blood cell is also known as a biconcave disc because it's a round disc that has two concavities on the side. It functions in oxygen transport. It has no nucleus. Our second formed element is a platelet or a cell fragment. It's involved in clotting or coagulation. Our white blood cells can be divided into two groups: the agranulocytes, which have no visible granules using a light microscope, and the granulocytes, which have visible granules using a light microscope. Our first white blood cell group is the lymphocytes. They include B cells, T cells, and NK cells, or natural killer cells. These cells have a large ovoid nucleus, small clear blue cytoplasm at the rim, and they function in destroying cancer cells, cells infected with viruses, and foreign cells. And some of them, the B cells, secrete antibodies. Our second agranulocyte is going to be the monocytes. They're called monocytes in the bloodstream and macrophages in the tissue. They have a large violet nucleus, a large cytoplasm, and they function in phagocytizing pathogens and dead neutrophils. And phagocytosis is just cell eating. And then we have our granulocytes. Our first granulocyte is going to be the neutrophils, also known as polymorphonuclear cells, or PMNs. They have reddish violet granules, and their nucleus is multi-lobed, containing 3 to 5 lobes, and they function in destroying bacteria. Our second granulocyte is the eosinophil. These cells have orange to pink granule. They have 2 large nuclear lobes, and they fight against parasites and pathogens. And our third set of cells are the basophils. They're called basophils in the bloodstream, and they're called mast cells in the tissue. They have a large nucleus that's S or U-shaped, and they have large granules, so many that they obscure the nucleus, and the nucleus is hard to see. These granules may contain an anticoagulant or a vasodilator. The anticoagulant is heparin, and the vasodilator is histamine. When you have an infection in your tissue, you want your white blood cells to migrate to that site. Usually, when there's an infection, there's damage to the blood vessels, and then coagulation will take place. You'll have hemostasis, you'll have constriction of the blood vessels, and you'll have a blood clot that forms. To prevent this, the mast cells secrete heparin to serve as an anticoagulant to prevent coagulation, and the mast cells also secrete histamine, a vasodilator, to prevent constriction of the blood vessel. Our second lecture on the formed elements. In the next lecture, we will discuss the size of these elements and their relative abundance within the bloodstream. And so that concludes this lecture. I hope that you've learned